What's up, biology students? It's me again. In our last video, we discussed how dominant and recessive alleles interact with one another to give us our traits. But there's more to genetic inheritance than simple dominant or recessive traits, and in today's video, we're going to look at a few more interesting patterns of inheritance that help us to understand how genetic traits are passed from one generation of organisms to the next. It's important to remember that this whole inheritance business is based on the fact that we have sets of chromosomes, and one chromosome in each set is inherited from our female parent, while the other is inherited from our male parent. That means that we have two copies of every gene, and may or may not have the same alleles for that gene. These alleles interact in different ways. Sometimes you need only one copy of an allele to display that phenotype or trait, and sometimes we need two copies of the same allele in order to display that phenotype. In our last video, we learned how to use these tools called Punnett Squares to help us predict the probability of producing offspring with a particular genotype. And based on that genotype, we can know what the phenotype or physical appearance of that organism will be. In this example we see here, we see that if two heterozygous yellow-seeded pea plants are crossed, there is a 75% chance that the cross will produce yellow-seeded offspring and a 25% chance that the cross will produce green-seeded offspring. This is because the gametes from each parent could combine in one of four ways, three of which produce a yellow-seeded plant and only one of which produces a green-seeded plant. And these are only the possible combinations of only one single gene. If we broaden our focus and consider the possible combinations of two single gene traits, we find that there are many more possibilities, many more combinations of chromosomes that could occur during meiosis and the production of gametes. Does this mean that if these plants produced exactly 16 offspring from 16 different matings, that exactly 4 of the offspring would be green? Absolutely not. It's just probability, or the odds that a particular phenotype will be produced. Just like you could potentially flip a coin 100 times and have it land on heads every single time, you could potentially perform this cross 100 times and produce 100 green wrinkled seeds. But that's just not very likely, not very probable based on the mathematical odds. But like I said earlier, this whole dominant and recessive traits business is only one way that alleles can interact. For example, how on earth can we explain this pattern of inheritance? If you remember, with our pea plants, it's not as though a green seed allele and a yellow seed allele produce a plant with seeds that are some color in between green and yellow, so what on earth is going on here? This is an example of what we call partial dominance, or incomplete dominance, which is pretty much just what it sounds like. The dominant allele is not completely dominant. In this scenario, a heterozygous genotype produces a phenotype that is kind of a blend of both the dominant and the recessive phenotype. In the case of certain flowers, if we take a homozygous dominant red-flowered plant, which has two dominant red alleles, and cross this plant with a homozygous recessive white-flowered plant, which has two recessive white alleles, we find that this cross produces only pink-flowered offspring. That's because these pink offspring inherit a dominant red allele from one parent and a recessive white allele from the other parent, because those are the only alleles that each parent could possibly pass on. They all have the heterozygous genotype, because this is the only possible combination according to our Punnett square. And for a partially dominant trait like this, the heterozygous genotype means a blended phenotype, a blend of red and white, which means pink flowers. Now suppose we take two pink flowers and cross those. Pink flowers, remember, are heterozygous. They have two different alleles for the flower color gene, one red allele and one white allele. This cross has a 25% chance of producing a homozygous dominant red offspring, who inherited a dominant red allele from each parent plant, a 25% chance of producing a homozygous recessive white offspring, who inherited a recessive white allele from each parent plant, and a 50% chance of producing more pink offspring, who inherited a dominant red allele from one parent and a recessive white allele from the other parent. And we can see these probabilities mapped out for us in our Punnett square. Some breeds of cattle display this same pattern of inheritance when it comes to their hair color. Just like our flowers, a heterozygous genotype produces a blended phenotype. So if we cross a homozygous dominant red-haired cow with a homozygous recessive white-haired cow, then the only possible combination of alleles is one dominant allele from the red parent and one white allele from the white parent. This heterozygous genotype produces a phenotype that in cows is called roan, which is a mix of red hair and white hair. This also means that we can never have a true breeding line of roan cattle, in which roan cattle who mate produce only roan offspring. Because if we cross a roan with another roan, there's always a 25% chance of producing a red-haired offspring, and a 25% chance of producing a white-haired offspring. In fact, we would only have a 50% chance of producing another roan offspring from two roan parents. There's another interesting pattern of inheritance when genes have more than one dominant allele. This is called codominance, because there are at least two alleles that qualify as dominant according to our definition. Our blood type is a codominant trait. The type A allele is dominant, and you only need one type A allele to have type A blood. The type B allele is also dominant, and you only need one type B allele to have type B blood. The type O allele, however, is recessive, and you must have two type O alleles to have the type O blood phenotype. But if both the type A allele and the type B allele are dominant, what happens if you have one of each? In this case, you literally would have both traits. You would have type AB blood. A person with type AB blood literally have both type A blood and type B blood. When it comes to red blood cells, blood type refers to the specific sugar molecules attached to the cell's membranes. These molecules, like many of the molecules attached to the membranes of cells, are called antigens because they may be recognized by certain antibodies as either foreign or part of the body. Antibodies are protein molecules, and they are a key component of the immune system. Antibodies identify potentially disease-causing agents, or pathogens, because those agents have antigens that the antibodies recognize as foreign. In the case of this figure, the shape and color of these little spikes represents the type of sugar molecule embedded in the membrane of these red blood cells. People with type A blood have the round purple sugars, or antigens, on their red blood cells, and people with type B blood have a square green sugar, or antigen, on their red blood cells. People who have type AB blood have both type A, or round purple antigens, and type B, or square green antigens. They literally have both phenotypes. People with type O blood, by contrast, have no antigens on their red blood cells, neither type A nor type B. People with type A blood also produce antibodies that recognize type B blood as foreign, because these antibodies are shaped so that they bind to the sugars or antigens on the type B blood cells. Likewise, people with type B blood also produce antibodies that recognize type A blood as foreign because these antibodies are shaped so that they bind to the sugars or antigens on the type A blood cells. People with type AB blood produce neither antigen, meaning their immune systems recognize neither type A antigens nor type B antigens as foreign. By contrast, people with type O blood produce both antibodies and recognize both type A and type B antigens as foreign. This is particularly important to understand when it comes to blood transfusions. Following major surgery and traumatic injury, people often require an infusion of blood to replace the blood that was lost. But give that person the wrong type of blood, and their immune system will recognize it as foreign and attack it. The host will reject the donated blood, making the situation potentially much worse. People with type A blood can accept a transfusion of either type A blood or type O blood, because neither type will have the antigens that the antibodies present will recognize as foreign. People with type B blood can accept a transfusion of either type B blood or type O blood for the same reason. People with type AB blood can accept transfusions from any donor because they do not produce any of the antibodies necessary to recognize the donated blood as foreign. On the other hand, people with type O blood can accept transfusions only of type O blood because their antibodies would recognize both type A and type B antigens as foreign. Type O blood can be donated to anyone because it has no antigens that could possibly be recognized by any antibody. So people with type O blood are referred to as universal donors because they can donate their blood to anyone. People with type AB blood are called universal acceptors because they have no antibodies to recognize foreign blood types. so they can take a donation from any blood donor. Let's consider a scenario. Is it possible for a woman with type A blood and a man with type B blood to have a child with type O blood? Well, if both parents are homozygous, meaning they have two of the same blood type alleles, then the answer is no. As you can see in our Punnett square, the only possible combination of gametes is one that produces offspring with type AB blood. But that should be obvious, because if neither parent has a type O allele to pass on, then it would be impossible for their offspring to inherit any type O alleles. The answer is also no if only one parent is heterozygous. In this case, even if the type A parent does have an O allele to pass on, the other parent does not, so it is still impossible for their offspring to inherit two type O alleles necessary to have type O blood. The same is true if only the type B parent is heterozygous. With only one type O allele between the two parents, it is still impossible for their offspring to inherit two copies of this allele. But if both parents are heterozygous, meaning that each parent has one type O allele to pass on, then there is a 25% chance that their offspring will inherit one type O allele from each parent and will have two type O alleles necessary to have the type O blood phenotype. Like many of our physical traits, our sex, whether we are biologically male or female, is determined by our genes. Specifically, our sex is determined by genes found on these two chromosomes, the X and the Y chromosomes. Females have two X chromosomes, while males have an X and a Y chromosome. Based on this configuration of chromosomes, we can use a Punnett square to help us understand why males and females are born in roughly equal numbers. If a female has two X chromosomes, then an X chromosome is the only kind of sex chromosome that she could possibly have in her gametes. But since males have both an X and a Y chromosome, then male gametes, or sperm cells, could have either an X chromosome or a Y chromosome inside. When the sperm cell combines with a female egg cell during fertilization, the egg cell's X chromosome is combined with either another X chromosome or a Y chromosome donated by the sperm cell. We can see in our Punnett square that there are four possible combinations of sex chromosomes that could arise after a sperm cell fertilizes an egg cell. Two of these possible combinations result in a zygote with two X chromosomes, and this zygote will grow up to be biologically female. The other two possible combinations result in a zygote with one X chromosome and one Y chromosome, and this zygote will grow up to be biologically male. It's a 50-50 chance, and that's why males and females are born in roughly equal numbers. Out of all 23 pairs of chromosomes found in a human cell, only these last two, the 23rd set, contribute to our biological sex. When it comes to genetic inheritance, this particular set of chromosomes is pretty interesting in a lot of other ways too. For example, the Y chromosome contains very few genes compared to the X chromosome. You can see from this karyotype that the Y chromosome is in fact much smaller than the X chromosome. Interestingly, though, although the X chromosome contains far more genes than the Y chromosome, the X chromosome contains very few genes that are directly related to sexual development. Since males have only one X chromosome, they also have only one allele for all genes carried on that chromosome. This makes the whole dominant and recessive alleles thing play out differently when we're talking about genes found on the X chromosome. In females, one X chromosome randomly shrivels up and becomes mostly inactive in all non-reproductive cells. And it's not even the same X chromosome that becomes inactive in each cell. Genes contributing to our ability to see in color are found on the X chromosome. Our eyes have these structures called rods and cones in this region that we call the retina that absorbs light and enables us to see. Rods just detect the presence or absence of light. They allow us to see the contrast between light and dark, but they do not help us to see any specific colors of light. Our cones, however, are sensitive to different colors. We have three kinds of cones: red cones that are sensitive to red light, green cones that are sensitive to green light, and blue cones that are sensitive to blue light. If one or more of these cones fails to develop properly, that can result in a condition known as color blindness. Red-green color blindness, a condition in which a person cannot tell the difference between red and green light, is caused by a recessive allele found on the X chromosome. This condition affects about 1 in 20 males, while only about 1 in 400 females. And this disparity is due to the fact that males have only one X chromosome, while females have two. In this chart, we can see how different phenotypes relate to the possible genotypes of males and females. As you can see, in females who have two X chromosomes, there are two possible genotypes that result in normal color vision, and only one that results in red-green color blindness. A female with this genotype, even though she is a carrier for the recessive color blindness allele, also has one copy of the dominant allele that results in normal color vision, so she will not be colorblind. A female must inherit two copies of the recessive color blindness allele to have this condition. In males, however, there is only one possible genotype that results in normal vision, and one that results in red-green color blindness. This is why red-green color blindness is so much more common in males, because with only one X chromosome, there is no chance of overriding a recessive color blindness allele with a dominant allele. One copy of the recessive allele is enough to cause red-green color blindness in males. Let's take a look at a few scenarios that illustrate why X-linked recessive traits are more common in males than in females. Suppose that a male with normal vision and a female who is a carrier for the color blindness allele have a baby. By setting up a Punnett square with the female's genotype on top and the male's genotype on the side, we can see all the possible ways that their gametes could potentially combine. Combinations will result in a baby girl, and two of these possible combinations will result in a baby boy. This is always the case. Whenever a sperm cell fertilizes an egg cell, there is a 50-50 chance that the resulting offspring will be male and a 50-50 chance that it will be female. But if we look at the alleles for color vision that are carried by the X chromosome, we see that if the baby is a girl, there's a 50-50 chance that she will carry the color blindness allele just like her mother. But there is 0% chance that she will actually be colorblind, because she cannot possibly inherit the color blindness allele from her normal vision father. If the child is a boy, however, there's a 50-50 chance that he will be colorblind, because there is a 50-50 chance that the X chromosome he inherits from his mother will have the color blindness allele. But boys do not inherit a second X chromosome from their fathers in the way that girls do, so this one recessive color blindness allele is all that it takes to make that boy child colorblind. In another scenario, let's suppose that a colorblind male and a normal vision female have a baby. Let's also suppose that in this case, the female is not a carrier of the color blindness allele. In this scenario, since the female has no colorblind alleles, and since boys inherit their X chromosomes from their female parent, there is no chance that these two parents could have a colorblind male child. They also could not possibly have a colorblind female child, since this female child would inherit one of her mother's two dominant normal color vision alleles. But since all females inherit their second X chromosome from their father, and since their father, in this case, carries a recessive color blindness allele on his X chromosome, there is a 100% chance that a female child produced by these parents would be a carrier for the color blindness allele. If a normal vision male and a colorblind female have a child, could their daughter be colorblind like her mother? The answer is still no, because if these two parents have a daughter, she will inherit one X chromosome from her father and one X chromosome from her mother. Her mother may be colorblind, but since her father is not, she will have one dominant, normal color vision allele, and she will also be able to see in color as a result. However, there is a 100% chance that she will be a carrier for the recessive allele, though, because she will have inherited one copy of this allele from her colorblind mother, who had two copies of this allele. If these two parents have a boy, however, he will definitely be colorblind, because boys inherit their only X chromosome from their mother, and since his mother is colorblind, she would have passed one of her two colorblindness alleles on to her son. The probability is 100%. So far, we haven't seen a single combination of parental genes that could possibly result in a colorblind female. In fact, the only way to produce a colorblind female child would be if both parents carried the recessive colorblindness allele. For example, if both parents were colorblind, meaning that the male parent had one copy of the recessive allele, and the female parent had two copies of the recessive allele, then there's a 100% chance that their children, boy or girl, will be colorblind as well. As you can see in our Punnett square, every possible combination of gametes results in either a girl who has two copies of the colorblindness allele, or a boy with one copy of the allele on his only X chromosome. And if the male parent is colorblind, with only one copy of the colorblindness allele, and the female parent is a carrier, not colorblind herself, but with only one copy of the colorblindness allele in her genes, then there is a 50% chance that any child conceived by these parents, boy or girl, will also be colorblind, and a 100% chance that a female child will have at least one copy of the colorblindness allele. So males are, overall, far more likely to have red-green colorblindness because males only have one X chromosome and need only one recessive colorblindness alleles to possess the colorblind phenotype. On the other hand, females must inherit two copies of this allele in order to possess the colorblind phenotype, and must inherit one from each of her biological parents. Haemophilia is the same way. Haemophilia is a bleeding disorder caused by a recessive allele carried on the X chromosome. People who have haemophilia lack the ability to regulate blood clotting, meaning that when they are injured, they bleed uncontrollably without forming proper blood clots to stop the bleeding. People who have haemophilia are far more likely to bleed to death from relatively minor injuries because wounds that should be able to heal themselves are not able to do so. As you can see in this figure, there are more possible combinations producing males with haemophilia than there are combinations producing females with haemophilia. And just like red-green colorblindness, males are statistically more likely to have this bleeding disorder than females, and it has everything to do with the fact that males only have one copy of the X chromosome. There's an interesting story about haemophilia in the royal families of Europe, actually. Historically, there has been quite a lot of interbreeding between the royal families of powerful countries in order to gain and consolidate power, form alliances, and in the pursuit of wealth and territories. This figure is what's called a pedigree. It's a sort of family tree that shows how certain genetic traits are passed through family lineages. We read this kind of pedigree from top to bottom, with ancestors at the top and their descendants below. This key shows us how to interpret this pedigree. Squares are males and circles are females. Red indicates the presence of the recessive haemophilia allele. Half-colored circles represent female carriers of the allele, and fully colored squares represent males with haemophilia. The squares with question marks are males who died in infancy, possibly because they had haemophilia, but that's not entirely clear. Is it also not entirely clear how haemophilia got into the royal families of Europe? As you can see in this figure, Queen Victoria of England, who lived from 1819 to 1901, is the first carrier of the recessive allele, and neither of her parents carried even a single copy of this allele. It is rumored that Queen Victoria's mother had a lover other than Edward, the Duke of Kent, and that this illicit lover had haemophilia and impregnated Victoria's mother and conceived the Queen herself. However, that is extremely unlikely, even if it does make for a good story, since males living with haemophilia at that point in history rarely lived to be old enough to even have children. So it's unlikely that this story of illicit love is how haemophilia got into the royal family. What is more likely is that Queen Victoria's recessive allele was the result of a random mutation that occurred at conception, and was passed from the zygote to every cell in her body, including her reproductive cells. This would make it possible for her to pass this mutation, this recessive allele, to her children. And if these children were male, they would not just carry the allele, they would have full-blown haemophilia. Such was the case with Victoria's son Leopold, who did live long enough to conceive children, and who passed this allele on to several of his descendants. Victoria's daughters, Alice and Beatrice, also each carried a single copy of the recessive allele. Through Alice's descendants, this recessive allele, and the bleeding disorder that it caused, was passed to the royal families of Russia, and through Beatrice's descendants, it was passed to the royal families of Spain. Understanding the patterns of genetic inheritance helps us to understand how disorders like hemophilia and colorblindness can be inherited by children from their parents, and how a great variety of traits, both beneficial and detrimental, can be passed through family lines, from parents to their offspring, and so on. These traits are determined by genes carried on chromosomes, and it is important to remember that processes like mitosis and meiosis play a huge role in how these chromosomes get passed from one generation of cells to the next via both sexual and asexual reproduction. And with that, I will bring our video to a close. Thanks for watching, and remember, you can go back and watch this video as many times as you would like until you feel like you understand the mechanisms of genetic inheritance and how partially dominant, codominance, and sex-linked traits are passed from one generation of organisms to the next.